Hi, welcome for today's session. And in this session, we are looking at, uh, basically, we are coming to look at lipids. We want to look at lipids, but in the previous lesson, we looked at something very important when we were looking at the structure of carbohydrates. We saw that carbohydrates normally are made of monomers, and these monomers are joined together to form long chains, which we are calling polymers. These long chains are called polymers. We saw that polymers of starch are branched, but those ones of cellulose are not branched. Today, we will go directly to the next unit that we are going to look at, and we will be looking at we will be looking basically at lipids. So lipids, as we know them, or if we have heard about them, they are somewhat different in structure as compared to carbohydrates, though they also provide energy just like carbohydrates, but they also have other functions. So lipids are non-polar molecules. Lipids are non-polar molecules. This non-polarity makes them difficult to dissolve in water. This makes them difficult to dissolve in water, and they include fats, oils, oils, and cholesterol. So these three are the main things that make up fats, and we might have also waxes and so forth. So some lipids are broken down as a source of usable energy for cells. Some lipids are broken down as a source of energy for cells, which means they are involved in cellular respiration. So other lipids are part of the cell structure. Other lipids are part of the cell structure. As we continue studying, we will be looking at examples of this, like the phospholipids and so forth. So fats and oils are two familiar types of lipids. They store large amounts of chemical energy in organisms. The structure of fats or oils are similar. They both consist of their structure. Their structure is similar, and that is, we are talking about fats and oils. Their structures are similar. They are made of, they both consist of molecules. They both consist of molecules. They both consist of molecules called glycerol. Bonded to molecules called fatty acid. Bonded to molecules called fatty acids, bonded to molecules called fatty acids. Many lipids, both fats and oil, contain 3 fatty acids bonded to glycerol. 3 fatty acids bonded to glycerol. So many of the fats and oils will be having that structure, and because of this structure of 3, we call them triglycerides. We call them triglycerides. So the name triglycerides comes from the fact that they have 3 structure. So we will be looking at this in details using our videos here. We'll be looking at this using our videos here so that it's somewhat clear for us. I will start with the first video here, and in this video, as you can see, what you see here is but examples of lipids. Lipids are normally liquid. They are normally liquid, though. So when we talk of triglyceraldehyde, we are basically talking about the 3 glycerol coming together in a bond to form something else. So we are having here examples of triglyceride, and then we have cholesterol. We have cholesterol. So they are very important. These are also referred to as hydrocarbons. They store a lot of energy, and that is why they are even able to burn to produce this energy. If we look at the structures, I said that 3 chains of glyceraldehyde, fatty acids, are linked to triglyceraldehyde. 1, 2, 3. These 3 form what we call triglyceraldehyde, and it's made of both fatty acids and glycerol. So these are the fatty acids, and these are the glycerol. So the fatty are the 3 molecules of fatty acids will join together with 3 glycerols to form these fats. Fats can either be saturated, like in the solid fats, or unsaturated. When they are saturated, it means that all the carbons, when we say they are saturated, it means that all the carbon molecules. So if we have it here, let me make an example here. So if we have carbon, then when we say it is saturated, we mean that all its 4 covalent bonds have hydrogen. And when it is not saturated, we might be having maybe a double bond with hydrogen molecules. So when it forms a double bond here, it means that one of the bonds is not saturated by hydrogen. So this is referred to as unsaturated, and this one is saturated. So we will be discussing how these are formed. We'll be discussing how these are formed. We have some other fats which are produced like oyster. This one is a human fat. It's not natural and it is very risky for our lives. Then we have phospholipids. Phospholipids are part of the cell structure and they are very important because they have chains which are both hydrophobic and hydrophilic. A portion of it attracts water molecules, that is the inside part of it. This side attracts water molecules, and the outside part is hydrophobic, which means it does not, it always repels water molecules. So this structure is very important for the function of a cell membrane, which is supposed to allow some materials in and also to stop some materials from entering. So let's look at how this happens here in a quick view of a video. Normally, what we are calling synthesis and hydrolysis of fats, we are having glycerol plus 3 fatty acids making a fat plus water. This is a dehydration synthesis. So here you can see that water is lost. So we have glycerol. This is the structure of glycerol. We have carbon-carbon molecules and OH groups here. As you can see the structure, the carbon-hydrogen, sorry for this. The carboxyl group, this is called the carboxyl group. This is the fatty acid. So typically about 16 to 18 carbon atoms long. So this is a fatty acid. We said 3 of them come together. So at least it will have one double bond. At least you'll have one double bond. So let's see what happens here. So this, sorry. So we are having here what we have here is a continuation. So fats can be saturated or unsaturated. So we have glycerol here, the structure of glycerol. It will need 3 fatty acids, and these are 3 fatty acids. So these 3 will come and join here together to form what we are calling fats and lipids. To call what we are calling fats and lipids. So as we progress here, it can be either saturated or unsaturated. So these hydrogen atoms come off during dehydration, and these OH groups also come off during dehydration to produce water. So if they come together, they will form, of course, bonds. They will form bonds, and water molecule will be lost. So as you can see here, this produces a water molecule, and this water molecule, and this one also produces another water molecule, and this one also produces another water molecule. So in this case, we are having 3 water molecules. So this one also produces another water molecule. as you can see there. So now what is formed here is what we are calling a fat or a triglyceraldehyde. A fat or a triglyceraldehyde. So it is very clear that when these two join, water will be released. Water will be released, and this is called hydrolysis of fats. This is called hydrolysis of fat. We can also put back the water to break the bonds. We can also put back the water to break the bonds, as you see here. So when we put the molecule of water, this will break the bonds. This breaks the bonds, very clear, and this will separate the, this is called a hydrolysis reaction, and in the hydrolysis reaction, water is put back, and the bond is broken, so that we end up with glycerol and fatty acids. Hope that is clear. We can proceed with our discussion. So as you have seen, that is how fats are formed or produced. So the double bonds on saturated fats make kinds in fatty acids. As a result, a molecule cannot pack together tightly enough in unsaturated fat. That is why normally it is in a liquid form. So the double bond in an unsaturated fat makes kinks in the fatty acid. This results the molecule that cannot pack together tightly enough to form a solid. So because of double bond, this one cannot form solids. Cannot. But on the other hand, if it is saturated, then it will easily form saturated fat is solid. It's normally solid in nature, and we call it saturated because normally all its outer electron bonds are saturated with hydrogen. The cell membrane is mostly made of a type of a lipid called a phospholipid. So phospholipid, unlike the first that we looked at, is typically somehow different. Unlike other lipids, phospholipid, unlike the other lipids, consists of glycerol, two fatty acids, and a phosphate group. So this phosphate group is very important for the structure here because it provides polarity, which is, of course, that is the part of the polar head So this is the part of the polar head, normally, of the molecule. So this side is hydrophilic or water-loving, and on the other part is hydrophobic, which means it is water-haring. It doesn't really attract water, and that is very important in this structure. Another one that we were to look at is cholesterol. So cholesterol, as we know it, is normally in excess or normally found in eggs and other animal fats. It's a lipid that has a ring structure. It is a lipid that has a ring structure. So it has a ring structure, and you may hear people maybe talking of the danger of eating foods containing cholesterol, such as eggs, but in reality, your body needs them. Your body needs cholesterol. It needs cholesterol for some functions which are very important. For example, cholesterol is part of cell membrane. It's part of cell membrane. It's part of cell membrane. So it makes part of the cell membrane. And also your body uses it to make chemicals called steroids, used to make steroids. And steroids as hormones are very essential. So cholesterol-based hormones that have many functions. These are cholesterol-based hormones that have many functions. Like, for example, they, for some, regulate stress. Some of them regulate stress. So we are talking about here the hormones. And others are essential for reproduction, for control of sexual development and reproduction. Control of sexual development and reproduction. Examples here include the testosterone and estrogen. So these two are responsible for sexual development, and also they are responsible for reproduction. In the next video, we will be basing ourselves or looking at the next important structure, and in this structure that we will be looking at in this case will be the proteins. For today's, we will stop here.